Hey, this is the video for Biodiversity.2. Go ahead and set up a new entry in your Cornell notes. Remember to start a new page for this video. Also check out the essential vocabulary. Today we will be discussing paper chromatography and gel electrophoresis. So paper chromatography, simply put, it is a procedure that separates pigments in a solution. This root word, this prefix chrome, means color. So we are actually separating the pigments in a particular plant, and this one was Botanicurus that you explored during the Relationships and Biodiversity lab. So the way that this paper chromatography works is you use something like filter paper. And very much like when you have, maybe you have like certain pens or markers, and when they get wet, they kind of smear, like if your book bag ever got wet and you see some markers or pens that are like smearing all over the paper. That's kind of what's happening in paper chromatography. So you add a drop of liquid of the pigment, you add it to the filter paper. You put this filter paper in water. Now this drop cannot be in the water or it will dissolve and it will diffuse into the water. So you want it to be above the water. So the water soaks up and then rises with it. And as the water rises up, it's dropping off parts of this pigment. So if we looked at this over time, the first thing that's going to happen is you're going to drop off the greens, for example. Then you'll drop off some bluish colors, and then some yellow colors, and then some orange or reddish colors. Now this is going to be different for each pigment. But this is the gist of it. This is the general appearance of it. Now, once this is done, you can compare your unknown sample with other samples. So if I had my unknown and I had samples 1, 2, and 3, I'm going to look at the one that has the most similarities to my unknown sample. And in this case, I have all of the colors present in number 3. So these two, the unknown and sample 3, would be the most closely related. And the reason for this is because they're going to have similar proteins. When they have similar proteins, it's because they have similar DNA. So the closer the DNA is related, the more closely these organisms will be related. Now, gel electrophoresis is a procedure that's using electricity to separate DNA segments based on size. Now, in this example, what we do is we take DNA and we use restriction enzymes. And restriction enzymes are basically like scissors. So we're going to cut this up into multiple segments. And it's going to cut all of the bases, all of the DNA segments. It's going to cut them in similar spots. In the lab that you did for living environment, it cut it between the AACC. So what you're going to do is you get all these little segments and then you put them into these wells. This is called a well, and this is basically a giant piece of gelatin. So imagine you had this piece of jello, and these DNA segments are different sizes because of the way they were cut. And these DNA segments are going to have to move through this gel. So what you're going to do is apply an electric current. And then what happens is once you have the electric current, these DNA segments move through this gel. And the larger segments get stuck near the wells, and the smaller segments will move all the way through the gel. So watch. And what happens is it drops off the segments based on their size. So as we go through this procedure, you now have different bands, and these are bands like a wristband. And what you do is you're comparing your unknown with your samples A, B, and C. So my unknown, what I'm going to do is I'm going to look at each one of the bands very similarly to how I compared the paper chromatography. So I have similarities there, similarities there. And as we go down, we want to look and see which ones are similar. Now, what I notice, I want to look at the one that has the most in common. It may not have every single band in common, and some you might see in different, in multiple segments or samples. But in this example, you have the most in common with sample B and the unknown. So the unknown is most closely related to whatever organism is represented by letter B. Now this can be used to again show relationships because similar DNA is going to make similar proteins, and similar DNA means that they are closely related. So I would like you to respond to this check for understanding. I want to make sure that you had a good grasp of what I was trying to explain. I thank you very much for your attention. I hope that this video was informative, and I will see you soon.